morning. Welcome to the Boardwalk Talks program of the Dauphin Island Sea Lab Aquarium. This morning we are coming to you live from Dr. Ken Hoadley's lab here at the Dauphin Island Sea Lab. Dr. Hoadley is going to chat with us this morning about reef-building corals and the algae that live in these reef-building corals and how they have symbiotic relationships. So with that, I am going to turn it over to Dr. Hoadley.
Thank you very much, and thank you very much for inviting me to this week's talk. So I consider myself a phycologist, which is just a fancy term for someone that studies marine algae. And one of the species that I like to work with is known as Zooxanthellae. And they're unique because they are found in a symbiotic relationship with corals. And so before going into the symbiosis, I wanted to take a second to talk about corals themselves. So perhaps some of you have been following some of the Zoom lessons that have been put out by Dauphin Island. And so my wife actually gave a talk a couple weeks ago on cnidarians. And so cnidarians are the sort of overarching group that encompasses jellyfish and corals and anemones. And so perhaps some of you are familiar with what a coral is already. But overall, corals, I have a photo from a coral reef in the Pacific up here. And they basically look like colored rocks. However, in reality, they are a type of animal. And so we have various coral species here, this one up top, different species down here, as well as over here. And if we were to zoom in on these guys, we would find that their tissue looks like this. And so it's a very thin tissue layer. And on that tissue layer, we have these structures. If you zoom out a little bit, perhaps. There we go. We have in the center here, we have a mouth, and it is surrounded by tentacles. And then if you look down here, we have this gray, or this brown material, and that's actually the symbiotic algae, Zooxanthellae, living within the coral tissue. And so this might look familiar because it kind of looks like an anemone, which they're closely related to. But on a reef, on a coral, there are colonial organisms. And so there's thousands of these little polyps all over the species you see up here. So each one of these species has hundreds, if not thousands, of these coral polyps. So corals make a living in two different ways. They can either grab food particles from the water column, so either phytoplankton or zooplankton, and they do that using those coral tentacles. The other way they can get energy is through photosynthesis from the symbiotic algae living within them. So the algae will photosynthesize, and they will produce this sort of energy-rich sugar, which is transferred to the host. And as the title of today's talk, how algae help make coral reefs, this energy-rich sugar is what helps corals calcify. And so their skeletons are actually what build the reefs. And specifically, symbiosis with these algae help them have enough energy to support the high rates of calcification required to build the reefs. So the corals that build the reefs are known as scleractinians. And globally, there's roughly 700 to 800 species. And then there's quite a lot of diversity of the symbiotic algae as well. So they're a little harder to tell different species because they all look very similar under the microscope. But through genetic analyses, we can tell that there's roughly 100 to 1,000 different species of Zooxanthellae. And so what I'm particularly interested in is the combinations of the host and the symbiotic algae, and how combined they elicit a certain response to either environmental stress. So perhaps you guys have heard of coral bleaching, and that's when the host coral sort of expels all of its symbiotic algae during high temperature stress. And the coral itself is left without its symbiotic algae. The algae is what gives it its tissue pigment, and so the remaining tissue is translucent, and what you're seeing is the underlying white skeleton. So that's where the coral bleaching phenomena comes from. as it turns out, some of the zooxanthellae, so the algae, are more tolerant to high temperature than others. And so there's quite a lot of interest in trying to understand why certain species of zooxanthellae are more tolerant than others. And that plays a big role in which corals we see bleaching. And so we're trying to figure out why and how that distribution is placed throughout the world. There are other environmental stresses I'm also interested in. So ocean acidification, which is a change in the ocean chemistry that can also impact calcifying organisms. And then perhaps more locally, nutrient regimes within the water column. So higher nutrients from human activities on shore can run off into the reef systems and impact what species of coral and the health of those corals as well. And so I'm very interested in how these different host and zooxanthellae combinations allow these corals to acclimate or better cope with these different environmental stressors. So how do I do my work? Well, there's two ways I can do it. I can either go into the field, and that's where all the fun photos come from, is diving on these reefs. And so there we can take samples and detect what species of algae are within them, or look at how an entire reef system is responding to a thermal stress event. But there are a lot of questions that require more nuanced approach. So there's experiments that we try to do in the lab. And for that, we need basically a model system. So similar to how the medical world has laboratory mice, coral biologists have model systems that we like to use in the lab as well. And so what I have here are tiny anemones known as Aptasia. And so for those of you that might have kept a marine tank in the past or currently do, these are the same little nuisance anemones that hobbyists are always trying to get rid of from their tanks. And they make a good model system for us for that reason. They're very hardy, they're very easy to take care of, and they quickly reproduce. So for these guys, you can see that some of them are browner than others, and it has everything to do with how dense the concentration of symbiotic algae living within them is. For example, in the center there, we have a very brown one, and next to it, we have a much paler one. And we can actually manipulate these to have them completely devoid of symbiotic algae. Next, we can infect them with different strains of the algae to see how the different species interact with one another to help the anemone adapt or acclimate to various environmental stressors. So this is a very useful model system for coral biologists. And the last thing I wanted to show you, because one of the things I'm particularly interested in is photosynthesis, one of the tools I use is known as a chlorophyll fluorometer. And this is a device that basically shines light at algae. In response to that light, the algae fluoresce, and the fluorometer detects that fluorescent signature. I can then look at that fluorescent signature and derive a lot of information about what's going on photosynthetically. So we can turn this on real quick. And you'll see we have a little bit of a light show with different colors flashing. So those are those high intensity flashes of light that are causing the algae within the coral to fluoresce. And you'll...
I'm sorry. We have a quick question for clarification.
Sure.
Liz asks if you would explain what you mean when you say infect them with algae. How does that work?
Right. So with a anemone that doesn't have any symbiotic algae within them, it's very pale. And we can choose different species of Zooxanthellae, which we place into the container with the anemone. The anemone then takes up those algae, and that algae grows a population within the host anemone. And so then we've reestablished a symbiosis within that algae based on a particular combination of symbiont and anemone of our choosing. So back to the fluorometer here, we see these different colors. And what's going on there is these different colors are exciting different pigments. So just like in the fall, where we see leaves in the trees turn colors, they're turning colors. The different colors they turn reflect the different pigments that they have within them. Similarly, algae also have different pigments within them. And using these different lights, we can see how those different pigments are being utilized for photosynthesis. So it's a really useful tool for that kind of analysis. It's also quite beneficial because I don't have to disrupt the organism itself. I can gain a lot of information with minimally invasive techniques.
So we have another request for clarification. When you say that they fluoresce, can you explain what that means? Or when you say that
So when they capture light, the algae themselves will emit a different wavelength of light, and that's known as fluorescence. And so that is something that we can detect and measure.
So when you use this fluorescent light, how do you tell that what you have done or what's happened with it?
So all of the measurements are being taken on a small microchip here, and after recording, I can then take that off and look at it on my computer.
So what kind of data do you gather in that?
I gather these traces. Basically, each flash of light is less than 1/1000th of a second long, and I can take about 100 or 200 fluorescent samples during that time. And what I'm looking at is a rise in fluorescence over that time. And the shape of that rise in fluorescence is what I'm interested in.
So is there, with the algae that you say you were infecting with, how is that what this is gaining information about, is seeing how that is infecting it?
It can. It's more looking at how the different types of algae in there photosynthesize. So certain ones are more efficient than other species. Certain ones have more of pigment A versus pigment B, and so that's the kind of information we're able to derive from this technique.
How many different species of algae are you looking at?
Right now, most of these have a single species of algae. So I just got these into the lab last week, so they're still sort of acclimating to their new environments. So I haven't quite gotten to manipulating them just yet.
So in the years of work that you've done with some of this, what is the most interesting thing that you've discovered along these lines?
Well, some work I did during my PhD is we looked at how these anemones consume zooplankton based on what algal species they had within them. We also had an anemone without any zooplankton in it. And as it turns out, the anemone without any zooplankton was least able to consume zooplankton. The ones with species of algae within them were much better at consuming zooplankton. So the idea there is that energy they're getting from photosynthesis helps the algae consume more through eating.
So you use the word infect, but the Zooxanthellae, the algae that is in the coral or the anemones, is actually beneficial to them, right?
Mm-hmm.
And did you find when you were looking at the comparison between the one that had no algae, no Zooxanthellae, compared to the ones that did, you found that that they thrived better with the algae.
Yep.
Do some of them have multiple species of Zooxanthellae, or would they typically only have one species?
So, yes. There are certain types of corals that are only found with one single species of algae. There are other corals that are found with several different kinds of algae. And as it turns out, depending on the stress, the environmental stress, or the environment in which it's in, say if it's very high on a reef where there's lots of light, or low on the reef where there's much low light, that can impact the relative amount of these different species of algae within the coral.
So it's more it's more influenced by the environment rather than the species of coral?
There are only certain associations that can be made for every coral, but then the environment helps dictate which ones are more dominant within that association.
So if they have a higher biodiversity, a higher diversity of algae, is that beneficial to the coral, or is that is there a correlation there, or you just see that they have more potential for multiple species depending on the environmental conditions?
That is a really good question, and very much right on the nose in terms of what people are studying right now. For a while, we've thought that having multiple associations is beneficial, especially during thermal stress. We see that more thermally tolerant symbionts become more prevalent on these corals during thermal stress. And so there's been this thought that that might be beneficial. There's more recent evidence that shows that corals that only have one species of algae actually recover from these thermal events better than those with multiple ones.
Interesting. Do you have any idea why?
You know, with these symbiotic algae, there's an incredible amount of diversity within them. So some of these different species have departed from one another, have evolved separately over hundreds of millions of years. So they're very genetically different from one another. And it likely has something to do with that diversity.
How many species are there?
Between 100 and 1,000, it's very tricky to know for sure.
And we have another question. Do corals die when they bleach?
They very much can. So if they cannot recover quickly, so they cannot reestablish that symbiosis, they can die fairly quickly. And if other stressors are involved, that also plays a role, and they can die even faster. It very much depends on where the coral is and what its environmental surroundings are during a thermal stress event.
So you mentioned a couple of other kinds of stress. And one of them was nutrients. So do you have a situation where the coral have these algal symbionts, and the algae photosynthesize, so they need sunlight in order to produce their food. Excess nutrients going into the water might affect them by supplying nutrients to other algal species that might create a bloom above them in the water column and shade them out, or?
So sure, nutrients can have all sorts of impacts within the marine environment. If we think about the impacts of nutrients on the reef itself, there are lots of different types of algae. There's macroalgae, which grows on its own, and are sort of
that seaweed.
We might call that seaweed, or certain types of seaweed. Those can benefit quite a bit from the higher nutrients, and so higher nutrients may make those bloom, which then might interact with corals and cause problems for those corals. Nutrients on these types of algae, so the microalgae, can disrupt the symbiosis with the host, and so that's how higher nutrients within the water column can also impact corals by changing the symbiosis they have with the coral.
So if we talk for a second about another stressor you mentioned, which was ocean acidification, just as a little explanation about ocean acidification, a lot of our viewers would have heard about excess carbon dioxide in the atmosphere, and that can result in carbonic acid rain into the ocean, and even a very slight change in pH, even a very mild acid. This is an experiment that people could do at home. If you took even a tiny little sliver of shell, a mollusk shell, and dropped it in a jar of vinegar, which is a mild acid, you could watch it dissolve.
Mm-hmm.
And then in the ocean, there are processes where this lower pH can be neutralized by dissolving calcium minerals. There's a cycle here, but there's some concern that organisms that have calcium skeletons that are easily dissolved in even a mildly acidic water can have some pretty serious effects from this more acidic rain.
Mm-hmm.
So can you?
Sure. So ocean acidification, as you pointed out, starts with higher CO2 concentrations within the atmosphere. Those high CO2 concentrations mix in the water and change how much CO2 is in the water column as well. What it generally does is it also decreases the pH in the water column, increases the CO2, and decreases a different carbon known as carbonate. Carbonate is important for calcifying organisms, and so decreasing that makes it harder for organisms to calcify. pH can have a number of, a decrease in pH can have a number of problems as well. A lower pH can impact how proteins within a cell interact with one another, and so that can be a problem for organisms. Lower pH can also, again, impact how organisms calcify, making it harder for them to calcify under those conditions. On the flip side of that, CO2 is required for photosynthesis. And so higher CO2 can benefit certain organisms. Corals are great examples because they have algae that photosynthesize using that CO2, but they also have a skeleton which is not benefited by ocean acidification. And so there's this interesting relationship as potential benefits versus decreases in pH and carbonate that can have real impacts on corals. So it's a very interesting area of study for corals as well as other photosynthetic organisms within the marine environment.
So Liz asks if you can show us how the anemones eat.
Sure. Let me
While you're grabbing that, we have another question. Is whether fluorescent has anything to do with the mineral fluoride?
It does not. Nope. So fluoride is a mineral, as you mentioned. Fluorescence here comes from the chlorophyll pigments within the algae. So what I have here are some Artemia. People also refer to them as sea monkeys. So little zooplankton. Normally we try to concentrate these a bit before feeding them, but in the interest of time, I'm just going to take a sampling. And then we will pour it directly on some of these anemones. And you can see their tentacles retracting as they grab some of these plankton and try to bring it to the center of their mouths.
So this is similar to what Logan chatted about when he talked about the corals in the aquarium that he cares for, of feeding them with the turkey baster for them to kind of grab with.
Absolutely. Yep. So
they're related to jellyfish, which people might be a little more familiar with.
Mm-hmm.
So they have that ring of tentacles in the mouth, right in the.
Yeah. Well, a simple way to think about it is, jellyfish are pulsing with the bell up top and their tentacles underneath. And if you flip that over, then you have your coral polyp with the tentacles up top surrounding a central mouth.
Liz said, they're so cute. Well, Ken, we appreciate the time that you've taken.
Well,
with
thank you
the
very much
Boardwalk
for
Talk.
inviting me.
And as you do your research and learn more, we'll share it with our followers and friends. And
As a follow-up to answer some of the questions that you are currently addressing.
Sounds great. Thank you.